This is part 2 in our 3-part series on ovulation, fertilization, and implantation. As the body approaches ovulation, the fallopian tube secretory epithelial cells become ciliated, and the suspensory ligament shown here on the right increases activity and lengthens as directed by hormonal signaling. When the ovum here on the left now is released from the ovary during ovulation, the fallopian tube elongates, and the fimbriae here at the distal end of the fallopian tube approximate the ovary. The fimbriae sweep over the ovary like fingers, kind of caressing and capturing the released egg during ovulation. When the egg is released from the ovary and picked up by the fimbriae, it then travels down the fallopian tube into the ampulla where fertilization is likely to occur. An ejaculate from an otherwise healthy individual delivers 200 to 300 million sperm into the vaginal canal. A simple schematic here shows the general structure of a sperm with its head that includes the nucleus and the acrosome, its midpiece, which includes the mitochondria and centriole, and a tail. We have gone over the structure of an oocyte in our previous video. Very few sperm will actually reach the ampulla, and about half of those that have made it have reached the wrong oviduct. Within the oviduct, sperm expose their acrosome and species-specific receptors, which draw them to the egg and allow for fertilization. It's important to note that species-specific receptors allow for only same-species breeding and procreation. Sperm are viable for 3 days in the ampulla, and when the oocyte is now in the fallopian tube, it releases a chemoattractant that binds receptors on the sperm and allows the sperm to identify and be attracted to its target cell. The species-specific zona pellucida receptors on the acrosome allow the sperm to penetrate the spongy corona radiata and actually bind the proteins on the zona pellucida. This is phase 1 of fertilization. Once binding of the receptors occurs, this triggers dissolution of the acrosomal membrane on the head of the sperm and release of acrosomal enzymes to clear a path through the protective zona pellucida. This is phase 2 of fertilization. Sperm that break through the zona pellucida attach to the cell surface receptors on the oocyte and initiates fusion of the cells to allow the sperm nucleus and midpiece, which includes the centriole and mitochondria, to be released into the oocyte. This here is an image typically taken from something like IVF, where you see a sperm getting through the corona radiata, about to enter and bind the zona pellucida and fuse with the oocyte inside. Once the sperm has actually penetrated the zona pellucida, shown here, and fused to the oocyte releases a wave of calcium and cortical granules into the space between the oocyte and the zona pellucida, called a perivitelline space. The cortical granules modify the zona pellucida, hardening it as to allow for no other sperm to enter and fertilize an already fertilized egg. This would be called polyspermia. Remember from our last video that when the secondary oocyte is released from the ovary, it is stuck in metaphase 2 of meiosis. With the fusion of the sperm to the oocyte, metaphase 2 of meiosis is completed, so only those eggs that are fertilized actually reach definitive gamete status. After fertilization, the egg and sperm fusion byproduct, now called a zygote, undergoes rapid cell division. It goes from a 2-cell stage to a 32-cell stage morula by day 4. The zona pellucida maintains a restrictive barrier around the developing zygote, so cell divisions don't actually expand the gestation, but rather subdivides the cytoplasm into smaller cells called blastomeres. These cells also secrete fluid, which form a cavity called a blastocyst, with a flattened layer of cells on the outside called a trophoblast. This is a primitive precursor of the placenta. Inner cells of this are called the embryoblast. This gives rise to the future embryo. And this is also an inner cell mass of cells which contains embryonic stem cells, a hot topic of research right now. Note here that polarity is formed at this stage, where you have an embryonic pole and an aembryonic pole. Around day 6, the zona pellucida breaks down and the blastocyst hatches from the capsule. This is a necessary step in implantation. The developing gestation enters the uterus as a blastocyst, hatches out, and the forming trophoblast extends villi projections to find endometrium and begin implantation. These are various photos of the actual hatching event taking place, where the developing gestation comes out of the zona pellucida. Typically, a developing gestation will implant into the uterine fundus or into the endometrium. This can be into the anterior or posterior fundus or endometrium. Issues here when we discuss fertilization really includes polyspermia, more than one sperm fertilizing an oocyte. Polyspermia can happen if a man's ejaculate contains too much sperm and can lead to some infertility issues. It typically leads to spontaneous embryonic death because extra chromosomes are not compatible with development.